Welcome everyone this morning to this fantastic session. My name is Guilherme Oliveira. I'm a researcher at the Vale Technology Institute in Brazil, and I'm very honored to chair this session this morning. We have an excellent lineup of speakers, and in general, they're going to tell you about what they're doing as far as personalized medicine and their own views on it. We had it eloquently put yesterday what's coming in this area, and I'm very happy to see that not only countries like France, UK, and the US are having their institute set up, but also Egypt has a very nice initiative going on. And obviously, you don't need to be reminded of the revolution that's coming our way as far as personalized medicine. That's basically taking a strong push from technology enabled by high-throughput DNA sequencing. I'm very excited to live in this era where these things are coming to fruition and actually making a difference in the lives of people who are ill, and also designing and describing to us what a healthy human being is. There are some very interesting projects that are looking at healthy people and all the variation that occurs in human beings from their own genome perspective, but also from their microbiome perspective. So this is really exciting times for this type of work. So the CVs of these very accomplished scientists are very long, so I apologize, but I'll make them shorter so that we don't take time from their own presentations. So our first speaker is Dr. Charles Offray from France. He is the president and founding director of the European Institute for Systems Biology in Lyon. He's a graduate of the University of Paris and the Pasteur Institute. He was a postdoctoral fellow at Harvard and a group leader of the Institute of Embryology. And he was the scientific director of the Gene Express program and head of the CNRS Research Institute in Molecular Genetics and Developmental Biology, then the Functional Genomics and Systems Biology for Health. Dr. Offray developed a systems approach to complex diseases, integrating functional genomics, mathematical, physical, and computational concepts and tools. He is the co-founder of the Image Consortium and the Systemoscope International Consortium with many other accomplished scientists. At his institute currently, he's fostering the transition from reactive to proactive systems medicine through the pioneers of health and wellness, including the Vistera project in partnership with ISCB and a large network of worldwide scientists. He has over 250 publications and many books and chapters and a highly cited list of publications. Today we're listening from him about how systems biology will revolutionize 21st century medicine through the Vistera project. Thank you, Dr. Offray.
Ana Farhan, Henimarco, Maratania Fimaxabet, Alexandria. I am pleased to be once more at Bibliotheca Alexandrina. I had the pleasure to be here 14 years ago when there was the opening of this marvelous facility. So today I'm going to present you the work we have been doing since then. Shortly after this meeting, we set up with two of my prominent colleagues. On the left, Professor Leroy Hood at the Institute for Systems Biology, and on the right, Professor Zhu Chen at the Shanghai Center for Systems Biomedicine, to rethink how to do our research for the goal of understanding life and improving health. 4 years ago, I moved to Lyon, and with the help of Lyon Biopole, a cluster bringing together academy, government, and industry, we kickstarted the formation of the European Institute for Systems Biology and Medicine, which was announced at BioVision in Lyon, and I'm pleased here to tell you about our progress since then. So 7 years ago, we kickstarted the field of systems medicine by launching this new journal, Genome Medicine, and you will find in those different references the summary of the situation as it was at the time of BioVision in Lyon in 2011. I'm going to tell you about the route we have been taking to develop a new approach to foster the transition from the current practice of medicine, which is only reacting when symptoms occur, to a more proactive practice of medicine that will emphasize participatory prevention upstream of personalized predictions when disease occurs. When we celebrated the 5 years of the journal, we were invited, Lee Hood and myself, to write an editorial on participatory medicine, and we emphasized the need to form teams between doctor and patients. And I will tell you now how we are going to do this and how we have started to do it in practice. So the first pioneer has been Michael Snyder, a professor at Stanford who endeavored to perform all the omics on himself and combine them with medical biological assessments. And in the process, he learned something that was happening to him, as you can see on the right panel, a sudden uprise of events that combined the onset of diabetes together with a viral infection. So he came back to his doctor, who recommended some medication and lifestyle changes. And he also came back to the lab with the hypothesis that maybe there could be a causal relationship between the viral infection and the onset of this complex disease. A second pioneer has been Larry Smarr, who spent about 20 years to look at himself through all devices that he could have access to. And in the process, he did two things: get a better health, as you can see from the transition from the left to the right, state of health. And he also could detect early signs of intestinal disease that escape classical medical examination, although he would not use any omics at that time. So 3-4 years ago, we decided to combine the two approaches into a single one. And of course, he had been looking at himself through nutrition, exercise, sleep, and stress, through a lot of what became known as a global area of mobile and connected devices. Indeed, we are witnessing the uprise of a large number of wearable sensors to collect all types of data on almost real time. It's true to say that there are like 40,000 mobile applications related to health that are available to look at sleep, nutrition, physical exercise, body and mind. As a result, we are embedded into a big data explosion, and we need to make sense of them while respecting individuals, citizen privacy, and even intimacy. So we went a long road already over the last 20 years. I'm highlighting here the main events and publications. You will find this in the PDF versions that will be distributed through the library later on. And then we formed a joint team with my colleagues in Seattle after we had discussed these ideas at our annual symposium in Lyon in June of 2013 and 14. We emphasized the real need for not only P4 medicine, but also P4 education. And then we launched the idea of having a worldwide joint effort, which is now known as the Vistera project. And I'm pleased here to be in Alexandria to highlight the progress we have made around the world to foster this idea. So the reason we can do this is because for the first time in human history, we are able to combine not only the classical biology and clinical features, the clinicome. We can leverage all the omics in an integrated manner, what we call the integrome. But we can also combine all these data in a meaningful manner to decipher causality from correlation through real-time measures of exposures to the environment and all those that Larry Smart has pioneered to measure, which we call the exposome. And by doing this, we can embark into describing trajectories between health and disease. What is really lacking here is a calibration definition of what is wellness, and not only what is wellness for entire populations based on averages, but what it is for single individuals. So we are going to promote the conduct of individual assessments over time of all the features that are listed on the right and the left that covers those three layers. And essentially we are going to ramp up to the entire human population, hopefully in the next generation, by conducting millions and then billions of such individual assessments over time that can be then combined virtually in the computer as new ways of conducting population studies. So in France, we have assembled a public-private partnership consortium to develop this idea. It's still in the early phase of development, particularly because of the sensitive issue of personal data protection regulations. And so we are now all set to kickstart this, to look at people in good health to start with, and of course, as soon as they enter into the risk area, to prevent them to develop disease, and whenever they develop disease, to make actionable recommendations to seek their doctor advice in order to prevent that to become very serious. Of course, we have to pick a number of those sensors. As you can see on the bottom of this slide, we are emphasizing training and education for people to use those in the best possible way. And for this, we are relying on the 40 years of experience of the particle physics facility, CERN, at the border of France and Switzerland. And this provides us with immense capacities for the long-term sustainability of the project. Of course, we need to have all these dense, dynamic data clouds not only connected, but we need to make sense of them. And by making sense, it's not only deciphering underlying mechanisms at the molecular level, but it's also translating this into actionable recommendations for all the stakeholders described at the bottom of this slide. And it goes from the nurse to the physical therapist, to the medical doctor, to the pharmacist, to the industrial developers, and very importantly also to the policymakers and politicians, so that we are really serving our communities. We face a situation where we are not only reacting when things go wrong, but we are spending 98% of our resources to react when it is most often too late, and only 2% to prevent things from happening. Although now, with those strategies and tools, real prevention is possible, as long as you can detect early signs of the transition from wellness to risk, to disease, and make actionable recommendations that can trigger the reversal of those trends early on if you manage to detect them, or later on if people are already engaged into the disease process. So across Europe, there are 20 of the 28 member states who have endorsed the agenda for personalized medicine. In already 12 countries, as you can see, we have people engaged in forming this consortium. And very importantly, if you look at the two ladies on the top of this, these are specialists of nutrition, because we believe nutrition is going to be a key player in defining what good health is. We now campaign to have a worldwide extension of this with my colleagues in the US and China. I was just a few days ago celebrating the 120th anniversary of Jiaotong University in Shanghai. We have also our colleague in India, Professor Samir Brahmachari, joining. And I hope through the library here in Alexandria, Africa is going to play also an important role. So we are at the very early stage of this. So far, only a few hundred of people have been monitoring this way. And we can tell you that we have learned a lot, not only on the logistics of this operation, but on the benefits that people can gain in a very short period of time. About 70% of them already after 3 or 6 months in the project, those that have been monitored in Seattle, have learned quite interesting lessons for themselves. And you will see Lee Hood presenting these results in conferences and very soon in publications. So we plan over the next 5 years to ramp up to 1 million people, which is at the stage of a rendezvous to check whether this is really feasible on the grand scale, and also whether we can bring down the cost in a way which is manageable, both in developed and developing countries. And if we are successful, and we think we should and will, every 5 years we will witness a ramping up of these numbers so that it becomes the new approach of medicine for the 21st century. Of course, we are not waiting for this to happen. We have been tackling already a number of complex diseases, mostly in the respiratory and cancer field, which are highlighted in these slides through the framework programs of the European Union. And we are now ramping up to foster the formation of this network of systems medicine center. Let me just provide you a few snapshots of new methods that we have implemented to make sense of these big data sets through these public-private partnerships. And one of them was dedicated to CV asthma. And you can see here a graphical representation of the complexity of the severe in red, mild in green, and non-asthmatic people through a new method derived from physics called topology data analysis, so which shows that combination of advanced mathematical and computational tools is really helping us to get a better idea of the underlying complexity of common disease. We are also developing a strong plan to have standardized computerized disease dynamic maps that goes from the wall chart cartoon representation, which are relatively static, to more dynamic machine and human readable format on the right. And we have done this already for Parkinson's disease at the Luxembourg Center for System Biomedicine. We are now developing an asthma map and soon maps for diverse types of cancer, starting with lung cancer and leukemia with our Chinese colleagues. And we have developed a platform called the eTrix Transmart platform that provides training and education in all these methods. It provides a standard starter pack to describe all the data types in a common format. It has also produced a very strong push on data curation, which is available publicly. And last but not least, I'm driving a work package on developing guidelines for not only recommendation but compliance with ethical, legal practice. And we have published a code of practice for the reuse of secondary studies of medical data, which is now being endorsed at the higher level by the European Union Data Protection Officer. To foster this transition from reactive to proactive medicine, we have been involved in the Coordinated Action CASIM to foster the implementation of system medicine in Europe. It has now created the European Association of System Medicine, which I'm chairing, and also a systems medicine hub, where you can find a lot of information of activities that are ongoing in Europe. We will have our first conference in Berlin next October. You can see some of the topics we are going to tackle. I'd like to emphasize that in this ongoing revolution, we need to pay attention to 4 important issues. One is to move from the classical, restricted, targeted, specialized informed consent to much more open informed consent that enables people to ask new questions to multiple data sets that we are not designed initially to address these questions. For example, to look at immunity and cancer in new ways. You need to have this informed consent in a more open format. We need to pay attention to harmonization and standardization so that we speak a common language. We also need to move away from the very restricted ownership of samples and data and even patients to a more open access custody of these samples and data. And the European Union has really established now a strong number of infrastructure to do this. And finally, we need to promote, as I said in the beginning, the formation of systems medicine centers, both in academy and industry across the world. For action without vision is only passing time, and vision without action is merely daydreaming. But if we combine vision with action, we can change the world. And I think Alexandria and its library and BioVision here is a good place to foster these important changes. This is a small team that comes from all over the world that helps me to develop this project. And as you know, it happened to us in France to be attacked. And as a reaction, I received a number of messages, including one from people here in Alexandria, and also a number of pictures that are highlighted here. So I'd like to close by thanking Professor Serageldin and the BioVision team for their message of solidarity with the French people on this occasion. Haheb Hash Korb, Professor Ismail Serageldin, وفريق البيوفيجن على رسالة الاتحاد مع الشعب الفرنسي. Thank you for your attention.
Thank you, Dr. Offray. We're open for questions. Is there a microphone? Well, I'll get to the crowd warmed up while the microphone goes around. So one of the things that I find very interesting at this time is that people are getting a lot of their own information, not only through Google searches, but also by wearing these devices that capture their own data. And they are getting empowered. So when they approach the physician, their relationship has switched a bit. I wonder what's your view on that and how the physicians are being trained to deal with this different situation.
So in order to tackle this very important issue, the Vistera project, and it has been really tested in real life now with a pioneer study in Seattle, will emphasize looking first at what defines wellness for single individuals. And in order to do so, we need to have a dialogue between individuals and wellness coaches. We are not medical doctors, but can translate the different types of data that we are collecting and the results of the analysis into something that makes sense to them in their real life, and therefore detect early signs of onset of disease that then will conduct individuals based on the recommendation of their coaches to talk to their doctors, having already processed their data in a way that they understand, and that forms the basis for a second dialogue between the patient now and his doctor. Of course, at the same time, we need to train the doctors from the early stage of the curriculum and those that are already in action to understand the meaning of the different types of data, because very often currently people go to the doctor and they know more about their disease than some of the doctors, because doctors have not been trained with omics or connected devices.
We have a question here from the table as well.
Thank you very much for the very interesting talk.
Dr. Ofe,
I don't know where it is.
over here, on the table.
Hey. I really like the recommendation from moving to individual consent forms to an open access consent forms, and also the free flow of information and data across countries. What you consider the main barriers to this? Are there any ethical barriers or logistical barriers that can get on the way of achieving this?
Well, the hurdles that we face are no longer scientific and technical. They are related to social acceptance and to legal issues. So I will start with social acceptance. It has to be bottom-up endorsement by citizens themselves across the world. And for this to happen, they need to trust that this is going to be done in a way that is respectful of their privacy and intimacy. Now, of course, we need to have a legal framework to do this. And currently, even in Europe, or even within countries in Europe, the ethical, legal regulations are implemented in ways that are very diverse, depending on institutional review boards in hospitals and patient organizations and many of the stakeholders. So therefore, we are going to propose very soon a charter for the World Alliance of Health and Wellness that will have overarching principles to foster a transition in law that enables this. At the same time, we are pragmatists, and we want to empower citizens with data exchange platforms that make this possible. And I know, and I notice that many of you in this country and in the worldwide audience have mobile devices like this. And you have to remember that 30 years ago, it was only a commodity for a few people. So it became a worldwide commodity because we form the GSM group that developed this data exchange standard for telecommunication. And together with CERN, who has developed the World Wide Web, we are working to develop the worldwide health, this platform of exchange that will enable anybody, wherever he or she is, to have access to this personal data and enable others to use it.
Okay, we have time for one more quick question from the audience. Yeah, we have one. I wonder if there's a microphone to. Where?
Hello. Are you concerned about the fact that by relying on wearable devices, there are many people who do that, but there's a very short time when they use it. I mean, there's a boom of that, but they use it for 3 or 4 months, and then how would you do that? I mean, because we also have been working on system medicine, and we encountered that problem in Venezuela and other countries where we work.
Yes, you are right. This is a barrier to implementation. It becomes fashionable. Everybody rushes into using it, and then people are not sustainable in their use of it. So this is where the constant dialogue every week, every month between the participant of the Vistera study and their coach is going to try to find the right balance of how much is it necessary to collect such data. So for example, you may need to do it only for 1 week a month to have meaningful data. So yes, so this is one important issue. First of all, how we manage the 40,000 and growing number of applications to only tackle a few, and then how we learn how to use them as much as is needed, but not too much. And this is the same for physical exercise or even for medications. You need to find the right balance. In other words, we need to take a systems approach to the overall organization of the process, and it is going to be a learning exercise between the participants and their coach.
And if I may allow another question, very short. I mean, do you use any kind of incentives for compliance? I mean, incentives to the users so they will comply with the measurements?
Well, I think the incentive so far has been through the dialogue between the coach and the participant to highlight what they have learned, which is beneficial for them in their regular life. And as a result, more than 90% of them are willing to continue beyond the first 1 year. And of course, as things consolidate, we learn more about the benefit for single individual, and we also ask them whether they like to share their experience with others. And little by little, it will form virtual communities that can share this data and redefine the trajectories from wellness to health to disease and back.
Okay.
So I will be here up to the end of the conference, and I'm happy to discuss further with many of you. Thank you.
Thank you very much, Dr. Foy. Okay, our second speaker will be Dr. Sherif El-Khamisi. He is a graduate of the School of Pharmacy here from Cairo University. He was a postdoc at the Genome Damage and Stability Center at the University of Sussex in the UK. And then he had some time working at the pharmaceutical industry in Egypt, and then moved to St. Jude Hospital in the US. Then he flew back to the UK, where he established his lab with a fellowship from the Wellcome Trust. He established also the Genome Center here in Egypt, and he's a fellow of the Royal Society of Preventive Medicine, and he won the Wellcome Trust Young Investigator Award. He's currently the director of the Genome Center at Zeeway City. He's the chair of molecular medicine and director of research at the Krebs Institute in the UK. And he's the recipient of the State Encouragement Award and Abel Hamid Shoman Award in Medical Sciences. His work is concentrated somewhat in DNA repair enzymes and repair mechanisms. And today we're going to hear his talk about genome diversity and instability and its relationship to precision medicine. Thank you.
Thank you very much. صباح الخير. أنا الحقيقة حابب أشكر في الأول المنظمين والقائمين على المؤتمر. أنا أول مرة أحضر البايوفيجن and it's just truly impressive. والتنظيم رائع والتوكس very high quality. فبحب أشكر كل القائمين والمنظمين على المؤتمر وعلى الدعوة لإلقاء المحاضرة هنا. I would like to thank the organizers for giving me the opportunity to present our work in this fantastic forum. So I will just start by giving you an outline of the talk today. So I will start with just an overview about what we study and what we are interested in the lab, with some brief examples of ongoing research. And then I will spin this to try to see where we're going in the future and the future of improving medicine. And I will focus on two things to make the therapeutics more precise or more personalized, which is to understand more about the genetic differences between patients and individuals, and also to improve some delivery methods. And I will just give you one example on the future of gene therapy. And then I will finish off with the big picture, how we see ourselves are and where we're heading in the future. And I would like to echo what has been mentioned yesterday about instability in general, which is we are what we are now because nothing is stable if you just give it enough time. So I usually give rather a comprehensive introduction about the importance of DNA repair, but I thought I don't need to do this again, because this was recognized this year by the award of the Nobel Prize in Chemistry to 3 scientists who discovered the importance of these mechanisms. And this is one of them. This is Thomas Lindell. And Thomas was the first to report and observe that the rate at which our DNA that encodes the blueprint for life decays at a very high rate, which is impossible and incompatible with life on Earth. And he reported this in Nature almost 40 years ago. And since then, people started realizing that there are mechanisms there that evolved in order to help cells and organisms to fix these rather high decay mechanisms of our genetic material. So this is the structure of the DNA, which you're familiar with, and these phosphodiester bonds are extremely stable, and the T-half of these bonds is millions of years. But the problem is because this genetic material lives in a very hostile environment with all the reactive species produced from normal metabolism, they constantly attack the genetic material and produce breaks. And not only this, also a lot of enzymes that metabolize the DNA structure during transcription, making proteins or a combination, they do so by breaking certain bonds in the DNA and resealing it very quickly. And if they don't do that efficiently, the cells will end up with breaks also in their genome. And people estimate about 7 lesions or 7 breaks in our DNA per cell per minute. And this is a very high number. If you consider in this 20 or 25-minute talk, in each of our cells in our body, we would have accumulated about 300 breaks per cell. This is a very high number. And if they not fix it properly, the consequences could either be mutations, because if these breaks are left and during cell division, the cell can start putting errors and mutate the genome, and that can cause, of course, all types of cancers. And on the other side, if the cell decides to be more wise and the breaks are very high, the cells may decide to die. And that can cause all sorts of degenerative disorders, specifically in the nervous system, and also during the aging-associated disorders, during the physiological decline of many organ functions during the aging. So the only happy outcome for the cell is to do properly, to fix these breaks very effectively and rapidly. So the tools we use in the lab to study these processes, we rely mainly on cell culture and primary cells or transformed cells and biochemistry, either pure biochemistry or things like chromatin immunoprecipitation, sequencing, and so on and so forth. But we rely also on model organisms. And we use yeast, the Saccharomyces cerevisiae, a lot, in order to mimic certain genetic pathways in yeast to identify new gene functions. And we have Minna, Sarafi, and Ahmed Awad in the audience. They're trying to manipulate the genetics of yeast in order to understand more about gene functions. We also use mouse. So we have Riham and Muhammad. Riham is currently in Germany trying to pick up some transgenic techniques in order to model some of these disorders in mouse. We recently used zebrafish, and zebrafish is a very good model because you can do high-throughput drug screens or genetic screens in basically a 96-well plate where you have one larvae per well. But perhaps the most rewarding organism, if I may say, is humans. And by this I mean we utilize the experiments conducted for us by nature and look for recessive disorders where mutations in certain genetic pathways that are responsible for maintaining gene integrity can underpin disease. And we have also Muhammad in the audience, and Wala, Wahiba, and Lamia. They're trying to utilize and look for these mutations and understand more about how they cause disease. So the breaks or the faults that happen in our genome come in many flavors. They can either hit one strand of the double helix or two strands. Sometimes you get cross-links between the two strands together, and sometimes you get base modifications also. And for each and every type of these repair events, there is a specific toolkit to deal with this. And the components of the toolkit differ quite remarkably, but also there are some similarities between these components. And the most important component in any toolkit is a Swiss Army knife. And the Swiss Army knife comes with multiple blades, as you can see here. And the reason for the multiple blades is because the chemistry, and the chemistry here is very important. The chemistry that arise at each and every DNA damage event, they are very distinct, and they need a specific blade in order to cut and replace the proper chemistry to bring back the DNA backbone together. So this is a scheme of one of the pathways where these red blobs denote the sites of the chemistry that are lost and needs to be brought back to the correct chemistry. The first step is for the cell to know that there is a problem, and this is, you need a goggle, and this is an enzyme called poly-ADP ribose polymerase, which jumps into these breaks and catalyze the formation of these polymers that basically highlight the problem and tell the cell there is a problem here, please come and fix it. And then comes after that this DNA and the processing step, which is very important. And there are many enzymes that are involved in this step. And the main function of these enzymes is to bring the chemistry back to a 3' hydroxyl and a 5' phosphate, upon which further repair can carry. And then a gap filling, and that is conducted by a DNA polymerase that leaves a nick, which is sealed by a glue, and in that case, it's a DNA ligase. So this is just a brief illustration of how the cells repair and fix these faults. Defects in this DNA processing step tend to underpin a lot of human diseases, and primarily the diseases that occur in the nervous system. So this is an MRI scan of 2 patients where they have defects in 2 different enzymes in this pathway. And as you can see, I, and I'm sure you, will not be able to distinguish between this scan and this scan, although they're coming from 2 different patients. The only thing in common is a degeneration of the cerebellum, which is this organ in the back of our brain that controls motor function and coordination. So that was very interesting to us, and we sort of set up to understand why the cerebellum is degenerated. And it turned out that the defects in this particular activity results in certain proteins being covalently stuck on DNA termini. So DNA is very long. If you stretch, it's about over 2 meters long. In order to compact it in a very tiny space of the nucleus, that creates a lot of supercoiling and catenations and compaction, which is a very smart way of doing it. However, it comes at its cost when the cell wants to do any sort of DNA transcription or replication or any DNA transaction. Then the cell needs to relax all these compaction back again. And this is done by these enzymes, which break 1 or 2 strands and allow the swiveling of the other strand around, the one strand or the other helix, the other duplex, in order to relax these tensions. And if these enzymes break, but they don't reseal, they become stuck, and that creates a problem for the cell. And of course, nature have evolved with another class of enzymes that liberate these proteins that are stuck on the DNA termini. And if these proteins and these enzymes don't do the function properly, you end up with human disease. So we set up to understand why the cerebellum, why this particular organ is affected by these mutations and these defects. And here we used yeast, we used a strain of yeast that we engineered to mimic this disease, and we did functional complementation. We asked what human gene can we put back in order to correct this disorder, and we found another activity, and this is really remarkable because it deals with exactly the same chemistry, but the chemistry on the opposite polarity. The first one was on the 3', this one on the 5', and that gives you an example of how specificity of enzyme action is really important. But the outcome at the end is the same. The outcome is a degeneration in the nervous system and the cells in our body that do not divide. And here we went back to the human models, and this is a family tree of an Irish family with 4 affected siblings, where we screened and identified by dense SNP genotyping. After that, we found a stretch of about 9 megabase with extensive homozygosity that was apparent only in the affected siblings, but not on their not affected sisters and brothers. And exome sequencing and Sanger sequence confirmed the mutation of this enzyme that we originally found in yeast. And that mutation, we then developed a biochemical assay in the lab to detect the activity of this enzyme. And what we look here is a band shift. If we see a band shift, that means there is an activity. If we don't see a band shift, that means there is no activity. And this protein also is gone, as you can see here, by immunoblotting. So clearly these patients don't have the activity. So we went back to the mouse model, and a collaborator of us generated the mouse model, and we did expression profiling, gene expression profiling using microarray gene chips. And almost 30% of the genes that were deregulated in the brain of these mice were associated with activities that maintain nervous system development and function. And almost more than 50% of those are associated with genes that are known to cause neurological disease in humans. So the model that we would like to put forward is these breaks, the protein-linked DNA breaks, are very common, and they are normal products of everyday life. Sometimes error happens, and if they're not repaired, they can affect nervous system function and development. And that's why we need these enzymes, that the main job of these enzymes is just to liberate these stalled proteins from DNA termini in order to bring them back to life. What we noticed when we did some proteomic analysis of these pathways is there are certain helicases, DNA RNA helicases, that tend to come together with these enzymes. And these helicases are, they really work with a structure, I'm not sure if you are all aware of, called DNA RNA hybrids. This is a DNA duplex, and together with it there is an RNA that is hybridized to it. They're called R loops. And this happens quite often during transcription because the nascent RNA tended to pair with a complementary strand of DNA during transcription. And that's normally not a problem if there are these helicases around because they resolve and they relax these pairing, inappropriate pairing. But if they're not around, they expose the non-template strand to breakage, and this is a major source of genome instability. So what we have in the lab, Reham developed some assays to look at these R loops, these structures, under the microscope to visualize them, to try to understand how they are formed, how they are repaired, and how they impact on human disease. Mohammed Ashur is an audience also. He's trying to understand the connections between these pathways and other pathways in the cell in sort of system biology type approach. These breaks also, when they progressively accumulate over time, without mutations, without pathogenesis, they tend to affect how our nervous system and other systems function over time, and they contribute to the aging or the premature aging process. And what we found is they suppress this somatotrophic axis by down-regulating the conversion of growth hormone into insulin growth factor 1, IGF-1. And they do so to promote somatic maintenance and suppress growth. And this is a very good hallmark for premature aging. But what is interesting in this is these breaks arise in the nervous system, but the conversion of growth hormone to IGF-1 happens in the liver. So that tells you that there must be some signaling process between the nervous system and the liver to trigger this down-regulation of IGF-1, which is important for how our cells cope with the aging process. So I will spend the remaining 5 or 10 minutes, I think, in how we utilize this knowledge in order to improve future medicine, or personalized medicine, or precision medicine. And the way we see this is utilizing differences in genetics between different patients, and also epigenetics, and to improve the delivery methods by gene therapy or other nano formulations, as you nicely heard from Dr. Mustafa yesterday. So I will just give examples on genetic differences and and utilizing these genetic differences either genetically or pharmacologically. So genetically, we are developing this gene editing tool, which is very recently identified by pioneering work for a number of scientists, where we can direct an endonuclease called Cas9 into a specific gene locus, which is guided by an RNA molecule, which is complementary to this gene locus. And that allows you to direct an endonuclease into a specific gene locus with very high precision. And the outcome of doing this is basically inducing a break, a double-strand break, a break in 2 strands of the double helix in one specific locus. And when this happens, there are 2 scenarios. One scenario is the cell tries to repair this DNA break by all the evolved mechanisms for dealing with this. But when they do this, they can either do this by a process called end joining, which is an error-prone mechanism. And when they do this error-prone mechanism, they induce deletions and insertions in this particular locus. And the consequence of these deletions and insertions in this locus is gene disruption. If you delete or insert certain areas in a gene, then you disrupt the gene and you end up with a knockout. And people use this mainly to induce knockouts, either in human cells, in zebrafish, in mouse, to model human disease. But what is perhaps more interesting is if the cell decides to utilize another pathway called homologous recombination. Don't worry about the names, but what you need to know about this pathway is it's an error-free, and utilize another piece of DNA from the homologous sister. But here we introduce with the Cas9 and with the guide RNA the bit of the DNA that is good, that is not mutated, and whereby we can replace the mutated copy by the good copy, if you like. So this is a very, very early stages, but we are attempting to establish the technology first with a view to hopefully do some progress on it. Now I will switch you to the pharmacological intervention. If you still remember this slide, the consequences of unrepaired faults in the genome, there are 2 bad consequences, either cell survival and cancer, or cell death and degenerative disease. But if you look at this bad consequence, which is cell death, if you consider this bad consequence from a cancer context, it can be a very happy outcome, because all you need for the cancer cell is to kill it, is to induce cell death in cancer. And that's indeed the case for the majority of chemotherapy and radiotherapy. Majority of chemotherapy and almost all radiotherapy. They do so by inducing damage in the genome, and when this damage reaches a certain threshold, the cancer cells die. So these 2 types of breaks where you have protein linked to the DNA, they are induced by drugs and medicine that is used in Egypt and elsewhere every day in the clinic to treat cancer. things like doxorubicin, daunorubicin, irinotecan, topotecan, all these chemotherapy, they do so by producing a lot of these protein-linked DNA breaks. What Muhammad Ashur and Delamy and the lab are trying to understand is they're trying to understand the genetic differences between patients that they take these therapies and how they respond differently to the treatment and whether this causes resistance or not. And they both proved me wrong to some extent. Is the genetic differences in many cases seems to be minimal, but what is important is the epigenetic differences. So that brings me to the concept of synthetic lethality. So synthetic lethality is best described if you look at a chair. If you have a chair sitting on 4 legs, and if you cut one leg, the chair is more likely to stay stable, more or less. But if you now cut 2 legs, the chair is more likely to fall over. And if you consider the cancer cell like a chair, it relies on 2 or 3 or 4 pathways for survival. If you know 2 of them, if you know one of them, then you're less likely to hit this cancer cell. But if you know 2 and you take away the 2, then you're more likely to kill it. And this is an elegant bioinformatics studies, and that brings me to the first talk and probably the following talk by Dr. Ahmed, where people did a whole genome sequencing of a variety of cancer tissues. And they mapped the mutations of a specific DNA repair pathways in each cancer type. And they found this remarkable distinction and personality in the cancer types. So they have this specific signature. So in acute myeloid leukemia, for example, you have this pattern where you have more mutations in this OD. OD is a DNA repair pathway. In P53, for example, in terms of ovarian cancer, and so on and so forth. So each cancer type has its own personality, has its signature in terms of mutation and in terms of defects that drove the cancer to occur. And the same for copy number variation also. So this is an example where breast invasive carcinoma, they have this signature toward HR. And HR is a homologous recombination pathway, which is utilized in this gene therapy approach by CRISPR. And that is being really utilized quite successfully in terms of synthetic lethality approaches. So if you still remember this enzyme that detects the break when they start, called PARP, poly-ADP ribose polymerase. This enzyme mainly detects breaks in one strand of DNA, in single-strand breaks. So if you have a single-strand break here arising and the enzyme PARP jumps into this in order to detect it and stimulate the fixation process, if you now inhibit this enzyme pharmacologically by designing a small molecules inhibitor to this enzyme, you can stop this step pharmacologically, so you can stop this process. But what happens to these unrepaired single-strand breaks if they're not fixed? If they remain, then they will, during cell division, they will hit a replication forks when the cell decides to replicate the DNA, and that can cause a double-strand break. And this double-strand break is repaired by this process called homologous recombination. And it turned out that one of the components, or 2 important components of this process, is 2 genes called BRCA1 and BRCA2, which a lot of you know about. And BRCA1 and BRCA2 underpin some sorts of inherited breast cancers. So if you now target this cohort of breast cancer, which do not have BRCA1 and BRCA2, and therefore cannot do homologous recombination quite efficiently, and this is a genetic defect, this is a genetic mutation. If you utilize this genetic mutation and now give them PARP inhibitor, now you can selectively kill these cancer cells. So these drugs are now, they've been through clinical trials, and they've been approved by the FDA late in 2014, and they have a very promising future in treating breast, ovarian, and a lot of other cancers as well. So the road ahead is to understand and utilize more about this synthetic lethality, and how can we achieve this is to adopt a system biology approach, but I'm focusing here mainly on the genetic and epigenetic differences. And the more we understand about the connections and the pathways and how pathways talk to each other, the more we can utilize this knowledge in order to tailor therapy to selectively kill a cohort of cancer in that case, or other diseases in a more precise way. So this is a big picture that I will finish off with. So we have lots of these breaks, oxidative breaks, and many types of breaks arise all the time. These breaks contribute to the nervous system function. If not repaired, they cause neurological disease. They also contribute to the aging process. They can trap proteins on DNA, and these trapped proteins are not good. They cause disease. And polarity here is very important. On this left-hand side, if it is on the 3' terminus, you end up with, again, a variety of neurological disorders. If it is on the other terminus, you also end up with a variety of neurological disorders. And the same process can be utilized in order to understand how cancer develops, which we're not so much interested in, but we're more interested in utilizing this knowledge in order to improve cancer therapy and response to therapy. And this is mapping of the champions of each and every process that we worked on. And I would like to thank the lab members, past and present. And this work is a collaborative approach between us and many, many other labs in Europe and in the US and in Japan. And this has not been possible at all if it is not collaborative effort. So I would like to thank everyone that contributed to this work, either directly or indirectly. We are funded at the Whale City by funding from the city, and by also funding from the STDF in Egypt, and by small funding from the EU, and the Wellcome Trust and CRUK. And I would like to thank you very much for listening.
Okay, we open for questions. So we have one question over there and another here.
Thank you very much for this very eloquent talk. And my question is about the future of the use of the CRISPR-Cas9. It's the specificity of the targeting. Is there any work ongoing in this area?
Yes. Yeah, this is a fantastic question. So this is a very, very infantile area. And in terms of the specificity, it depends mainly on the PAM side. So if you engineer your guide RNA, which is preceded or followed by these PAM sites, so far people think that it can be very, very specific to this particular gene locus. But the sequence of this PAM site, there is a debate in the literature whether you need a couple of Gs, for example, on the 5' terminus or not. So this is still work ongoing. But it's very promising. People have developed a lot of animal models using the CRISPR technology, mainly zebrafish, and it appears to be specific enough. Now, the big question is how you make it tissue-specific. Not just gene-specific. And that brings back to the idea of gene delivery and delivery by things like gold particles that Dr. Mustafa talked about yesterday. Or what we're trying is trying to engineer a tissue-specific promoter upstream of this Cas9 thingy, which will enable the expression of the endonuclease only in certain tissues. For example, the brain or pancreas, whatever tissue is. So that's how you can achieve this tissue specificity, not gene specificity.
We have another question here to the left.
I'm interested in knowing how did you make the link in the first release between the neurodegenerative disease and the mutations in TDP1 and TDP2? How did we make this link in the first place?
Okay. That's a very good question. We didn't make the link. We didn't make it at all. So how it arises is there was a paper in Nature Genetics that reported a mutation in this enzyme in one of these disorders. And 6 years later, we studied this enzyme from a pure biochemical angle. And we found that it actually repairs these kind of breaks. So we made this assumption that defects or accumulation of these breaks can be the cause for this neurological disease. And that's how the whole story started, by making a mouse model, doing studies in undividing cells, and so on and so forth. That's how it started. The other enzyme started by a very basic genetic screen on yeast, not on humans at all. And the human connection came later, about 3 years later.
Any other questions? I have a quick one. I may have missed on your slide, but how come this defect manifests in the cerebellum only?
Yeah, this is a great question. So the short answer for that is we don't know.
Okay, yeah, fair enough.
That's the short answer. So we tried many approaches, and each approach we tried, it didn't work, in a sense that it didn't answer our question, but it led us to other new avenues, which actually are more exciting. But we don't know why the cerebellum is affected, but people think that it is affected because this is an organ, the only organ in the nervous system that continues developing postnatally. So it continues to develop about 1 month after birth. So that can put the cerebellum at more risk of exposure to all the things that can damage the genome. And that could be why the cerebellum is defective. Another possibility is maybe this, again, genetic redundancy. Maybe the cerebellum, other tissues have other parallel pathways that can adapt and adopt to deal with these breaks and fix them, but the cerebellum have only 2, for example, instead of 4. So it's more reliant on these pathways in order to fix these breaks. And that brings me back to the genetic and epigenetic differences. But we still don't understand why the cerebellum is affected here, why the motor neurons are affected in things like motor neuron disease, ALS. We don't know this selective vulnerability of tissues, and the nervous system is largely poorly understood.
Okay. Thank you very much. So we are unfortunately out of time, but I'm sure Dr. Akamizi will be around to answer more questions. Thank you very much for your presentation.
Thank you.
So our final speaker for this morning session is Dr. Ahmed Mustafa. He's currently an associate professor at the American University of Cairo. His training was in bioinformatics in the Department of Biology and Biotechnology graduate program. He's a computer science, graduated from the Faculty of Engineering in the university here in Alexandria. Then he joined the Arab Academy of Science and Technology. And then he worked for Kaiser Permanente in Pasadena, in California, as a senior software engineer. Then he crossed the street. Actually, before that, he went to work for the PhD program in the University of Iowa. Then went back to California as a postdoctoral fellow at the J. Craig Venter Institute in San Diego, California, where he worked on metagenomics and metatranscriptomics. So his work varied from microorganism genomes to metagenomes. And today he's back in human work, and he's going to talk to us about the 10,000 Genomes project.
Good morning, everyone. First, I would like to say that it's a huge honor to be standing here today, joining such a wonderful crowd of audience and speakers. I would like to thank the organizers, especially Dr. Rami Aziz, Isra, and Marwa. Today I'm going to talk about what we have been working on for the past year or so at Human Longevity, and I'll highlight what would make a genome a clinically relevant genome. I'll give you an overview of what we saw in 10,000 genomes. That's the largest population of complete genomes so far. So we should all be familiar with that picture. Our bodies are made of tissues made of cells. Cells contain the nucleus, and the nucleus holds our DNA, and our DNA is made of chromosomes, and the chromosomes are the ACTG. 23 of these chromosomes make our entire genome of 3 billion base pairs. A single change in this base pairs, ACTG, could lead to dramatic effect on our condition of health or disease. To be able to detect these changes, we need to genotype, meaning that we need to sequence or we need to figure out the condition of the nucleotide A, C, T, or G and compare it to some reference. To genotype, there are different methods. We'll go over that. And to have some statistical power to assess the change compared to the reference, we need to genotype population. So we need to genotype and associate with a phenotype in a population domain. The different approaches to assess the genotype are SNP arrays, whole exome sequencing, and whole genome sequencing. SNP arrays have been contributing so much to the genome-wide association studies, but they, even though the most dense SNP array is 2 million markers, it still only covers 0.01% of the entire human genome. SNP arrays also suffers from an issue that it's based on population studies. So SNP array that was based on a report from a European study doesn't really fit with other like African population. Whole exome sequencing, it's based on sequencing approach, but it also covers only the coding regions of the genome, which is only 2% of the entire genome. The other issue with whole exome sequencing is that most of the SNPs that are associated with diseases are related in non-coding regions. So whole exome sequencing, which is focused on the coding regions, but most of the SNPs that are related to diseases are non-coding regions. So the alternative approach is to do whole genome sequencing, which covers the entire genome. These are the most recent papers published on genome scale sequencing over the past 2 years. And as you can see, the coverage of these papers from Japanese, Iceland, the UK, African, they do not have deep coverage. They claim to have about 6 or 7 or 10x coverage of the entire genome, but that doesn't qualify them to be clinically relevant genomes. What would make a genome to be clinically relevant? It has to be depth in coverage, meaning that every single position in the genome is sequenced multiple times. So when you call a position A, C, or T, or G, you have some confidence in that position. It needs to be coverage in breadth, meaning that it's covered most of the genome. And it needs to be high quality and reproducibility, meaning that if you sequence the same genome over and over again, you see the same nucleotide at the same position over and over again. How can we achieve that? So the cost of sequencing has been dropping down over the past years. If you remember the 2001, that was the first human genome, which was funded by the US government. It cost $100 million US dollars. Now we are approaching to the scale of $1,000 for a single genome. That's thanks to the next generation sequencing platforms. That's an overview of our workflow for doing the sequencing at HLI. So the subject or individual comes in. We get the DNA from the blood cells, and we put it on the sequencing machine. It's an Illumina platform. The sequencing takes about three days for one sample, and after that it gets uploaded to the cloud. We use the Amazon cloud for all our computing infrastructure. The bottlenecks or the most consuming time points are the sequencing itself, which takes about 3 days, and aligning the data generated by the sequencer to the reference genome, which takes about 1 day. That's an overview of how our sequencing covers the entire genome. So the black chromosomes are the reference, and the blue bars are how much we cover out of that genome. So you can see we cover about 85% of the entire genome by our sequencing platform. Also, we cover about 92% of the coding regions and 96% of the clinical relevant variants. If you compare that to the whole exome sequencing, so on the top panel, we have 85% covered by whole genome sequencing, and on the lower panel, we have about only 2% by whole exome sequencing. By sequencing the 10,000 individuals, we found about 150 million variants in the entire population of 10,000. We added about 6% to what is already published in DBSNP. So that's the addition we created from 10,000 population. And we expect that when we reach the 100,000 individuals, we will have about 500 million SNPs. That means that we're going to see a SNP every single nucleotide in the genome. So we have 3 billion base pairs, and we're expecting to see one variant, one mutation every 6 nucleotides. Not only we found new mutations in the 10,000 individuals, we also found fragments of DNA, whole fragments of DNA that were not reported before. The total size of those fragments out of the 10,000 individuals is about 3 meg base pairs. Each individual comes into the sequencing, generates a number of unique variants that haven't been seen before. The average number is about 8,000 variants. The largest number of variants contributed for the first time by an individual comes from the African population. So an African individual contributes about 14,000 new variants that haven't been seen before. European individual would contribute about 7,000 variants that haven't seen before. We have seen 150 million variants. Most of those variants are rare, meaning that they are not shared with most of the population. And however, those variants that are rare are very common. So the frequency of the variants on the x-axis here, we see the rare ones, the not frequent in the population, are very common among the entire population, but the variants that are common are somehow rare. So rare is common, and common is rare in terms of the frequency of the variants. Why rare variants are important? So here is a case where we have a variant that is specific not only to the family, but it's specific to the child. So we have that boy, he suffers from Dillman syndrome. It causes some semi-solid cysts that get filling the cavities in the skull, back in the skull, as you can see here, inside the skull, and in the eye cavity. We sequence the genome of the mother, the father, and the affected son. As you can see from the sequence data, the father and the mother are homozygous, similar to the reference, meaning that they do not have the mutation. But right here for the case of the affected child, the child has a gene that got mutated to A, and so the child himself is heterozygous. The father GG, mother GG, and the son is GM. So it's a rare mutation, but it has a dramatic effect on the child, on that child. So we wouldn't be able to detect such a change, such a mutation, without deep coverage. So we have a deep coverage. We can for sure say the homozygosity of the father, the homozygosity of the mother, and then the heterozygosity of the son, and the de novo mutation in the son. What is the rate of mutations across the entire genome? This is chromosome 6, just an example to show you across the entire chromosome. Each dot represents the number of mutations in 1,000 base pairs, a wind of 1,000 base pairs, and on the average, we have about, let's say, 60 mutations every 1,000 base pairs, and the higher the dots indicate a higher variability region on the genome. And if you remember from Dr. Ismail Sarageddin yesterday's talk, that's C4, which contributes to schizophrenia. It belongs to an area where we have actually high mutation rate in that region on the P arm of chromosome 6. If you break down the genetic variant, the rate of genetic variant across the different features in the genome, so we have the coding regions, and we have the non-coding regions from ENCODE, the regulatory elements, and we have the intergenic and origin of replication. So the most conserved area of variation is the alternative exonic, and the least conserved genetic region is the small non-coding RNA. Also, the enhancers are very conserved, and the promoters not so much of conserved in terms of variability. On the average, each individual comes in. Again, we contribute about 8,000 mutations, but in total, the total number of mutations or variants in that individual is about 4 million mutations. And those are the single nucleotide variations. And there are about 600 that are structural variations. And by structural variations here, we simply means the small indels, small less than 20 nucleotides insertion or deletions. If you look at the correlation between the single nucleotide variations and the structural variations, you can see here there is a somehow correlation between the SNVs and the SVs, meaning that the more SNVs we observe, we can see also more structural variations. If we color those, stratify those individuals, so each dot here is one person. If we stratify those by the ancestry, we can see the Africans up here, so and the Asians down here, European in blue, and the admixture in green. So we can see, based on the ancestry, the number of variations varies dramatically. The most variable genome are the Africans, and the least variable genomes are the Asians. If we look at the variability by gender, males versus females, and we stratify by the age. So if we look at the age on the x-axis and the number of variations on the y-axis, and here are the females and males on two different panels, you can see the trend of the variability of over age by females. It doesn't change much, but the variability by age for the females is actually increases, meaning that males accumulate more mutations when they grow up compared to the females. It's not very clear so far what it means, but simply it means that females, they can maintain their genomes better than males while aging. I'll move to another component, which is the 3D structure of the DNA, the non-coding component of the DNA. Here we have the worm and the human. The total number of genes estimated in the worm is about 19,000 genes, and the number of genes in the human is about 19,000 genes. So we can simply say that the number of genes doesn't correlate with the complexity of the organism. So the number of genes doesn't actually explain why human is more complex than the worm. So where does the complexity come from? The complexity comes from gene regulation, and there are different ways to regulate gene: transcription regulation, post-transcription regulation, epigenetics, and the recent technique, recent mechanism, which has been being appreciated as a way to regulate gene regulation, is a 3D structure of the genome. So the genome is not just a DNA within the cell, but it's broken down or grouped into what is called topologically associating domains, the TADs, and those TADs are sort of insulated from each other. So here is on the left panel, we have two TADs, TAD in green and TAD in brown, and there are barriers between the two TADs. That barriers is contributed by CTCF binding site. The barrier makes sure that the regulation on the left, on the green domain, the enhancer, cannot regulate the gene on the brown domain. It can only regulate the genes on the green domain. If a mutation happens in the barrier, if a mutation happens in the CTCF binding site, that wall is broken down, and that would affect or allow the enhancer on the green domain to regulate the gene on the brown domain. A paper in Science that got published 3 weeks ago showed that if you break down the barrier between two domains, you can actually contribute to cancer in 3 or 4 different types of cancer. One of those cancers was TCR enhancer. One of the enhancers from one domain got activating a gene LMO, leading to leukemia. Here I'm showing you from the 10,000 population, from the 10,000 individuals that we sequenced, how the variation of the variants across those TADs, across these topologically associating domains. So here we go from the positions on the TAD, from the left border to the right border, and in the middle. And the y-axis shows the number of mutations that are clinically severe or pathogenic. So you can see clearly from that chart that on the borders there is an enrichment for the variants that are pathogenic, and the variants in the middle of the TAD or the topological associating domain they are not that pathogenic. The last component of the talk is the unmapped genome. So not all the reads that we sequence from an individual, they get mapped to the human reference genome. On the average, 1 billion of the reads, 1 billion of the fragments, 1 billion of those sequences get mapped to the human reference genome, and about 50 million reads do not get mapped to the human reference genome. So we thought to examine those reads that do not get mapped to the human reference genome and see what they are. We first focused on the viral matches, and we found some significant hits against viral genomes. The most abundant viral component was human endogenous retrovirus K113, and if you know, that virus represents about 8% of our human genome. So it's part of our genome, but it's not yet part of the reference genome, and again it represents about 8%. So it wasn't very surprising to see a high level of K113 in the population, or in the 10,000 individuals that we sequenced. But what was interesting is that we saw it change somehow, or dynamics in the load of that virus in the population by age. Again, on the x-axis we have the age going from very young to older people, and each point is an individual. And the y-axis is how much we see of that virus of K113. If we smooth the points, you will see the trend going from high to lower to high again. If we group those individuals by decade, every 10 years, you will see more clear the pattern of the abundance of the viruses. It starts high, it goes down, and it goes up again. The explanation for that, we assume that would be the hypothesis for us, is as early on, the infants, they do not have a strong immune system, so we see much of activity of K113, and then the immune system starts to kick in, and as the immune system gets stronger, so it can repress the activity of the virus, and then the immune system gets weaker, and we observe higher activity of the virus again. Again, that's speculation. We're not sure yet if it's true or not, but that's what we have from our data. At the very end, I would like to summarize the results. We generated the first 10,000 deep coverage genomes with clinical standards. Again, deep in coverage and breadth in coverage. Identified 150 million variants. Identified an average of 8,000 variants unique for each individual comes in. Formulated a profile to identify pathogenic versus non-pathogenic variants in each individual. Found the relationship between the pathogenicity of the variants and the location on topological associating domains, and detected some dynamics or activity in K113 across the age of the human. And at the end, I would like to thank you so much for listening.
So we were told to cut short our time here, but I'll give you one question. I would have myself many, but one quick question so we can wrap up the section.
It's a fantastic talk, thank you. So there was a paper that seems to suggest that all the genomes of our European and American colleagues have African traits, and seems to suggest that everything is actually originated from Africa. What's your comment on this?
By African, you mean African Americans?
Yeah.
Yes. So everything is related to Africa at the end, but definitely there was a bottleneck after going out of Africa, and that bottleneck led to reducing the heterozygosity in other populations compared to the African. So as you can see, the African population has higher number of variations, higher heterozygosity, but after going out of Africa, the Asians and European went through bottleneck, and that limited to the number of variations in the population. So what we see in European and Asians is very different from what we see in the Africans, simply because of the out of Africa theory and the bottleneck that these populations went through.
Well, I'm sure that Dr. Mustafa, as well as the other speakers, will be around and happy to answer any other questions you may have, so please approach them. I encourage you to do that. And thank you again for your very interesting presentation. Let's thank Dr. Mustafa and the other speakers for this session.